What we're going to talk about now is the development of B cells and T cells, and recognize that a lot of the details of this particular section are tested because we have several different clinical correlates related to them. For example, if you're trying to look at a patient who has some kind of a hematopoietic cancer, say, we can do some immunostaining and determine whether or not that particular cell has certain markers that would be specific, for example, to a B cell or a T cell, or maybe a myeloproliferative type cell. So that's why these particular markers that we'll discuss are important. And then the other aspect of this is to why we focus on some of these details is because there's a number of pharmacological treatments now that can target these drugs, or rather that can target these particular markers in order to treat various types of blood cancers. So let's talk about, first of all, B cells. Now, recognize that we only have, 20,000 or something like that genes, right? But yet we have a B cell repertoire, if you will, of something like, 10 to the 10 different types of B cell receptors. And when you say B cell receptor, it means that a B cell receptor that can recognize this many different things, whether it's going to be a pathogen or maybe it will be some kind of an antigen. Let's say, for example, that I'm blood type A, and if you're blood type B and you give me your blood, then of course I've got a B cell receptor that is going to recognize and I've got antibodies against you. So it's all kinds of different antigenic determinant that will be able to recognize all these, 10 to the 10. So how do we do that? Is how do we get that diversity with regard to B cell receptors when we don't have that many genes? And that's essentially what the development of the B cell that we're going to talk about. But first, when we think of a B cell, let's think about it this way, is you've got a B cell. Okay, and remember, if you go back to the big picture, hemopoiesis, IL-3 is specific to your lymphocytes or your myelocells, okay, so neutrophils. But it's the IL-7 that's specific to your lymphocytes, so your B cells, your T cells, and your NK cells. So that's what's the first thing is that this is the cytokine that's going to be specific to development of B cells. The second thing is, where does B cell development happen? Where is it? It's in the bone marrow, stays in the bone marrow. And so that's where the story begins. Now, the other aspect of a B cell is that you've got your basic B cell, and it has a B cell receptor. The B cell receptor is the same essentially as what eventually happens is you get plasma cells, okay, and plasma cells are going to secrete antibodies, okay. So these antibodies are the same essentially as this B cell receptor. So just that's the sort of relationship. If you have a B cell that encounters an antigen and it processes it, okay, this is a B cell, he processes the antigen. And remember, the B cell can then present to a T cell, okay, and we'll go into more of that in another section in terms of antigen presentation to the T cell. So this particular guy here will eventually become specific, the same specific antibody. So for example, if I have a B cell that's going to recognize Epstein-Barr, and I'm going to produce a bunch of antibodies for Epstein-Barr, this particular type of Epstein-Barr, this particular flavor, if you will, then you're going to have these antibodies that are specific. The relationship there is the same. Maybe that's clear to you. I didn't really grasp that, and it was kind of a source of confusion for me. So let's talk about how this whole process works in terms of B cell development. So bear with me. We have a pro-B cell, okay, so pro-B cell, and this is about the development of this guy right here, okay. You've got this this portion here, but this is all about this development and specificity and diversity of this B cell receptor. Okay, so you have the pro-B cell and you have the pre-B cell. All right, and then you have the immature B cell and the mature. And, or some people call it the naive B cell. So where does the story begin in terms of being able to recombine and shuffle the cards on the genes? Okay, so I'm not a molecular biologist, but I'm just going to outline how it's typically tested for a beginning or medical immunology class. So we start with what they call the RAG1 and RAG2. Recombination RAG1 and RAG2 is going to be in this pro-B cell. Another way that you can do it is you have this particular guy here, this TDT, which stands for, which is going to be your terminal deoxynucleoside transferase, and that's another way to essentially insert your nucleosides into this equation to shuffle again the cards with regard to this receptor. And you're also going to get your V and J variable and junctional region. Again, you've probably already covered this in class. We're just kind of going over, the big picture here in terms of some of this VDJ recombination. And then you'll also have this CD79 A and B, which is a transmembrane component, okay, in here. But the important thing to remember is, first of all, what's important is RAG1 and RAG2, okay, is recombination. It happens at the pro stage. In TDT, you know, if you see this enzyme or this particular mechanism, that you're dealing with the B cell, and it's also true for T cells too. Again, that's something that they look at in terms of cancer and trying to figure out if a particular cell is of which type of lineage, be it lymphocyte or myelocyte. So that's something that you might see tested, okay? So this is important. And very important here is you get a marker called CD19. Cluster designation 19, and I consider it almost like just a cluster designation is almost like there's some bioactive obviously component to it, but I think of it like a little name card that it's got on a cell. So if I say to you that, for example, you've got a cell and all it stands for is CD19, you know that it's a pro B cell. If it doesn't have CD20, okay? So all B cells will have CD19, even the pro-B cell lineage. CD19 and 20 are specific to B cells. And when you're looking at cancer, I always think of, one way I heard of it was if it's a 2-digit, you're talking about B cells. If it's a single digit, generally you're talking about T cells, which we'll get into CD2, 3, 4, and 8 as we go into T cells, okay? So in this portion, you have allelic exclusion. What does that mean? That means that once you get a recombination that works in this particular receptor, then let's say from your mother, for example, and that gene rearranges efficiently, then you don't have genes from your father. It's not like the HLA molecules where you have co-dominant expression, and that's what allelic exclusion. And you get somatic recombination. Somatic, because these are somatic cells, recombination of your heavy and light chain, okay? Heavy and light chain. And we'll talk more about the structure of the receptor itself, but suffice it to say, you know, you've got your heavy and your light chains with regard to the antibody, which again is the same as, essentially the same structure as the as the B cell receptor. And then the next thing is, in terms of immature B cell, is you have tolerance induction, okay? Now this is important because if the cell is tested and it's autoreactive, okay, then it can either just die by apoptosis or it can become anergic, A-N-E-R-G-I-C, and in which case, you know, it's not going to be active. So that's tolerance induction. We go a lot more into that with T cells, but suffice it to say it happens at the immature B cell stage. And then the B cell receptor, this is in the mature stage here, okay, and so at this stage, you're going to have the IgM, okay? So you're starting to finally develop something that looks like what we're really interested in, identifying antigens. This happens to the IgM, and then at this becomes IgM, and then at this stage, of course, you carry on the IgM Oh, and you have IgD. Okay, and remember, IgD is never secreted, right? It's never the right answer, as they like to say, but it is a component of this mature or so-called naive B cell. All right, and so that's the function here, and you also have this molecule, okay, which is CD40. Now, let's just review and highlight. CD19 is pro-B cell. All right, CD20 is at the pre-B cell stage, and then at the mature is CD40. CD40 is exceptionally critical because it is the counter molecule to your T cell. This is a T cell who has a CD40L. Okay, so the T cell, she has CD40 ligand, or that's what I call it anyway, and then the B cell has the 40. If you don't have this interaction, you will not be able to switch from an IgM. So all you'll have is an IgM. So they call it hyper IgM syndrome. Okay, so you're not going to have IgG, you're not going to have IgA, you're not going to have IgE. So that's hyper IgM. Is if you don't have this CD40 molecule, and we'll go more into that in immunopathology. So that's a critical component. You also have alternative splicing, okay? And that's just, again, a function of shuffling the cards. And they're not going to go too much into the details of that, but the way that it's typically tested is they'll say alternative splicing, where is it in the stage of development, and it would be a mature. Or they'll say tolerance induction, where is it? It's in the stage of an immature B cell. Or they'll ask you, where is the receptor, what stage is it? Where is IgM only? It'll be in the immature stage. But the ones they really love, because we have a drug, a beautiful drug that targets CD20, okay, for people who have B cell cancers, B cell lymphomas. So those are the kind of questions that are typically asked. Let's just make sure that I didn't miss anything significant. Of course I did. So in this stage right here, all right, because again, a lot of these things come down to immunopathology, either too much or not enough. Okay, so in this stage right here, all right, there's an enzyme called BTK, Bruton tyrosine kinase. All right, so if you don't have this BTK, you actually stop development at the pre-B cell. So you don't fully develop beyond pre-B cell. So you don't have a lot going for you in terms of B cells. Okay, now, for all of us who took biochemistry, we always think, well, if there's an enzyme, it won't go beyond this stage. It won't go beyond pro. No, this is immunology, not biochemistry, even though there's a lot of correlates. So essentially, it doesn't properly develop past B cell. Now, there's some other mechanisms, they call it B cell leakage and some different terms, where you might say, oh my gosh, on a test question, the patient does have some B cells, but yet everything points to the fact that he has a BTK deficiency. That's okay. Just recognize that it's not fully functional, okay? It's not a fully functional B cell complement. And remember that B cells, going back to what do we need them for, all right, is they're very particularly instrumental in bacteria. So if you see a patient with a lot of bacterial infections, then you want to think, maybe it's a B cell. If you see a patient with only a certain type of bacterial infections, like a pyogenic bacteria, then you might think of something more innate, like NADPH oxidase deficiency. Okay, so that's how you want to differentiate in terms of immunopathology and immunodeficiency. So that's pretty much it on the B cells. Now, I'm going to ask a few questions combining B and T cells, but recognize that a lot of people will just say, well, the Tcell receptor combines very similarly to the Bcell story. Okay, so it's just sort of a hand-wavy type of a mechanism, but the T cell is very similar. Okay, and we'll talk a little bit more, about the structure of the antibody and get into the kappa and lambda chains and all that, but not at this particular stage. Now let's talk about T cells. All right, so the T-cell receptor, in terms of its recombination and shuffling the gene cards, again, you've got somewhere in the neighborhood of, let's say, 10 to the 11 different types of T-cell receptors, and it goes through a very similar mechanism that the B cells do. So we won't go into that. But the thing about the T cells, a couple of different points that I've seen asked a number of times, is that you have different kinds of T cells, okay? They go from the bone marrow, end up in the thymus for maturation. And in terms of the types of T cells that you might have, is you might have what they call a gamma delta T cell, okay? Or you have an alpha beta T cell. Okay, and these are your CD4 and your CD8 positive T cells, all right, as well as your Tregs are gonna, you know, the Tregs are also coming from the thymus, okay? These are very unique, and there's a subject of a great deal of research right now. They go out into the liver, they're various parts of the body, but when you see a test question asked, and they may ask you, you know, they'll say that it's CD4 positive, and then they might ask you, is it an alpha beta or is it a gamma delta, and it's going to be an alpha beta T cell. Now the story with regard to T cells is that, which we'll go into a lot more in terms of antigen presentation, so I'm not going to go into the as much of that, but in terms of the cluster designations. But remember how in the B cells you had your CD19, your 20, and 40 were kind of the big cluster designations with regard to B cells. Okay, but with T cells, single digit, you have CD2, which are going to be on all T cells. You have CD3, which is a T cell receptor, and then you have CD4 positive or CD8 positive, okay, one or the other. And remember that CD4 positive cells are the helper T cells, and those become activated by MHC class I molecules that are either on dendritic cells, or they could be on macrophages, or they could be on B cells. Okay, so that's who gets these CD4 cells activated. And they call this an MHC class II restricted cells. And the reason is because a CD4, okay, positive T cell, remember, has a T cell receptor. And it's good for seeing 10 to the 11 different types of antigens. But it can't be activated just by encountering some antigen that it happens to see. It's not, and it has to be presented an antigen by one of these types of cells in order to be activated, in order to secrete its cytokines, in order to proliferate the adaptive immune response. All right, and then there's CD8 positive cells. These are your cytotoxic T cells, which we've talked about already, and those are going to be activated based on MHC class I. Okay, so they come out there, they're smart, okay, and smart cells, and they see antigen presented in the pocket of an MHC class I molecule on a poor cell that's been infected, and then they're going to kill that cell dead. So you can imagine that these are quite instrumental with regard to killing virally infected cells. All right? So that's the story in terms of the T cell designations, in terms of the cluster designations. Again, remember the T cell receptor, 10 to the 11th-ish, and that goes through the recombination very similarly to the B cell receptor. So if you see staining for TDT, okay, like we talked about with regard to B cell differentiation, you know it's going to be a T or a B cell. Okay, so then they say, oh, it's CD4 positive, you know it's a T cell cancer. So that's where the clinical correlate with regard to that is. Now, in terms of maturation, okay, so you have the thymus, and you have the cortex, and you have the medulla. All right, and in the cortex, what happens is the T cells get put through the paces. All right, so what they do is, the idea is they want to see Okay, if the thymus wants to see if that T cell is going to react appropriately, are they going to be too reactive, not reactive enough, or just right? It's almost like a Goldilocks situation, okay? So what they do is they get exposed in the cortex to MHC class I molecules on thymus epithelial cells. All right, and so if they get one that's like, you know, he's all over the poor little MHC epithelial cell, then it's like, okay, negative selection. You're out of the pool. You had too much reactivity. You could potentially attack your own. So they call that negative selection. If, on the other hand, the little T cell, he goes in and he's just like, you know, looking around, he's scratching his back or whatever. There's like, then in that case, it's called death by neglect, okay? Sometimes we feel that way in medical school, but at any rate, it's death by neglect in this case because he wasn't reactive enough, and so he gets put out of the pool. Now, if it's just right, so in other words, he recognizes the T cell, recognizes the MHC molecule on the thymus epithelial cell, but not overreacts, then it's positive selection. So he makes the cut. Okay, it's positive selection. I think they say it's something like 1 out of 50 or something. It's probably better than the acceptance rates for medical school. But at any rate, it's positive selection. Now that T cell gets to come in to the medulla. Okay, and then he really gets put through the paces. So now he goes through a process involving the air protein. Now it's not the cheap old, you know, MHC class I off the epithelial cell. Now it's this very interesting protein, the air protein. And the way that I liken that is well, let me just say that what it is. It exposes that T cell to every type of tissue in the human body. I mean, it's phenomenal that we have this kind of capability. And so that T cell has the opportunity to react, to not react, to overreact, or to react just appropriately to basically every different type of cell in the body, okay? And if he doesn't make that test, again, negative selection. But if he makes that test, then he gets to become a fully fledged, fully functional T cell, okay? And so you might well imagine that if you have a defect, you as a person has a defect in this air protein, that means that you never got to put your T cells through their paces. And as such, you may have very reactive T cells, okay? You may have a lot of so-called immunopathology, overactive immune system that doesn't properly activate or, recognize self from non-self, okay? So we went back to it. Cortex, positive selection, negative selection, death by neglect, okay? Medulla, positive selection, in which case it's exposed to the air protein. The other component that comes out of the thymus are the Tregs, like I was mentioning, that you want to have regulation, even though those T cells have gone through in essentially a tolerance factor to make sure they're not going to start activating on your own cells. You still want what we call these sort of policeman cells, these Tregs straight out of the thymus. And these cells, if you look at how their little calling cards, if you will, are going to be CD4 positive cells, so they're alpha beta T cells, CD4 positive. They also have CD25, which is just another word for an IL-2 receptor that other T cells have as well, obviously. We'll talk about the cytokines in the next one. And then FOXP3, which is a transcription factor for the regulatory cells. Now, the confusing aspect of this is these Tregs come out of the thymus, but there's also Tregs that are out in the periphery that are made or differentiated out of your other types of T cells, which we'll go into that when we talk about cytokines. And the growth factor for those guys, and they're called AI Tregs, are TGF-beta, which is another cytokine. So they look the same in terms of, their complement of markers, but they're not homegrown from the thymus. So that's a common question. AI regs are out in the peripheral and TGF-beta. So let's go through some questions on B and T cells. The first question is, what marker do all B cells possess? This is kind of a tricky question because it's saying all. So we're talking about all the B cells, pro-B cells, pre-B cells, mature, immature, etc. So the only one that answers that correctly is CD19, which is A, the first question. Of course, CD20 doesn't show up until the pre-B cell stage, as you recall. So that's why A is correct. In terms of question number 2 is, what marker do all T cells possess? And that's CD3, right? Because you have some CD4 positive, some CD8, but CD3 is the T cell receptor marker. Question number 3 is, what is expressed on the B cell that has undergone isotype switching to IgG? We talked a little bit about that, but again, it's just essentially what marker is, and it's the IgG marker, and that's C, IgG B cell receptor. Point being is that the B cell receptor and the immunoglobulin, you want to think of those two things being the same. Number 4 is, at what stage in B cell development does tolerance induction take place? And that answer is going to be A, the immature B cell stage. Number 5 is, where does central tolerance take place in the thymus? And so central tolerance is going to be in both the medulla and the cortex. Remember that it's 2 processes that we went through. First, you go through positive selection, negative selection. Death by neglect can be the first sort of layer with regard to MHC class I epithelial cells. That's going to happen in the cortex. And then those who make positive selection go to the medulla, and that's where they're exposed to the air protein, which shows them all the different tissue types of the body. And at that point, they become fully mature T cells if they make it through that. And then number 6 is, a double positive T cell has the appropriate avidity, will undergo what? And again, that's going to be positive selection. So those guys are double positives, meaning CD4, CD8 positive cells. Number 7 is, a mutation in the following protein will result in autoimmune regulation defects. And that is going to be, as we discussed, A is the air protein, because that's the one that shows the T cell all the different tissue types of the body. If it doesn't have that, it will be autoreactive potentially. B, Bruton's A-gamma globulinemia. Remember, that was a defect, happens to be X-linked, and that essentially stops the B cell development. So you had pro, and then you had pre, right, and then onward to immature, and it's right here is BTK, but it stops it at the pre-B cell development, such that you don't have B cells, so you're probably going to be subject to bacterial infections. The C is the C1 inhibitor, INH. So remember, that one is, if you have a deficiency in C1 esterase inhibitor, then you're going to be subject to edema, aka angioedema, swollen lips, and close the throat, and that would be obviously lethal in that case. And then D is, if you didn't have Tregs, what we're talking about there would make that correct, is if we had said, if it was a FOXP3 deficiency or mutation, that, remember, is an integral part of the transcription factor for Tregs, so that would make that correct. But the correct answer, autoimmunity with regard to regulation defects, is going to be the A, the AIR protein. D wasn't specific enough, wasn't the most correct answer. And number 8 is, a mutation in what will result in low or dysfunctional Tregs, and we just answered that question. That would be A, FOXP3. If you look at B, we talked about air protein. C, AI regs. That's the guys that are out in the peripheral, same markers, but different growth factor, TGF-beta, not grown within the thymus. And then DAF is that going to be the decay activating or accelerating factor, and that is going to be part of complement regulation that prevents that process from acting on your cells and taking it through the membrane attack complex and blowing up your cell through autolysis. The next question is, which of the following T-cell regulatory mechanisms at the level of the antigen-presenting cell T-cell interaction is regulatory? And the answer, I'm just going to say it, is going to be B, and we're going to go into that more when we get into antigen presentation. So that's CD152 CTLA-4 on the T-cell, and then the other one is with the antigen presenting cell, which is CD8086 or B7. And again, we're going to go into that in an entirely different section. Last question is, which of the following T regulatory agents is derived from the thymus? We've talked about it 100 times, and that's C. The Tregs themselves come straight out of the thymus, CD4 positive, CD25, aka IL-2 receptor, and FOXP3. Thank you.